Welcome to this edition of Research Connection. Joining me today is Dr. Ben Callahan. Thanks for joining me.
Thanks, Kyle. It was an excellent prompt.
Thank you. So you recently got a couple of grants, correct?
I did, yes.
Tell us about it.
So I recently got 2 grants, so one from the USDA, one from the NIH. And I guess I'll talk about the NIH grant because it's more money, so it's more important. That's the way it goes, right? But so this grant is an R35, and so this is kind of like an R01 in that it's similar amounts of money, but it's more investigator focused. And so what we wrote about, or what I wrote about and am funded for, is really building on my past research for the past 5 years. And it's related to, I would almost call it metrology, or the science of measurements. And so what we're interested in is really quantitatively understanding measurements of microbiomes, so measurements of complex microbial communities. So that sounds a little dry, and it is, but really accurate measurement is sort of at the heart of a lot of scientific advances, right? And so if we can take a measurement that is true, then we can do things that we just can't do, even with well-designed experiments, even with fancy statistical analysis, if we have systematically skewed measurements or biased measurements or just inaccurate measurements. And so that's what we want to confront in microbiome science, which is, in some sense, it's been revolutionized by measurement technology. So it's been revolutionized by the advent of high throughput sequencing and other newer high throughput omics technologies that allow you to characterize a whole community sample all at once. So it's changed, it's sort of invented the microbiome field. Not that people didn't study microbial communities, but were able to do it at a greater scale, and as part of that it got this new name, microbiomes. But these measurements are not accurate. But the problem with these measurements of microbial communities is that we don't even really understand how they are wrong, at least not quantitatively. And that's for a couple of reasons. So one reason is that it's difficult to take those often short sequencing reads and say with high accuracy exactly what the bacteria, what species the bacteria was it came from, or exactly what gene it came from. So that's one challenge. But another challenge is that some bacteria are easier to measure than others. And so all of these steps introduce this systematic skew between the measurement and the truth. And so we've kind of referred to that as metagenomics bias, and our goal, our biggest goal with this NIH grant is to solve that problem. And our approach is to quantify it, so to develop mathematical models that relate exactly the measurement you get to the truth in a community, to validate those models, to determine where those models truly describe the difference between our measurements and the truth, and where they might not do as well. And then to use those mathematical models to guide analysis that might be insensitive to bias, to evaluate how robust or not robust our analysis of microbial communities might be, given that there is bias. And then third, the big thing that if we achieved would be, a notable achievement, but is going to be hard, is to develop calibration for microbiome sequencing measurements. And so that's the big picture goal of this NIH grant. And I won't bore you with the details. There's a lot of practical challenges in making all that happen. But that's why they gave us the money to try and do it. And if you're interested in learning more, we have published the first paper on that in eLife just like a month ago.